Okay, thank you, Winda. Okay, thank you, Miss Yulian Dianti. Next up, please welcome the coordinator of Biology Study Program, Dr. Reni Indrayanti, Magister Science. Time and place are yours.
Thank you, Auriga. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, and good morning to Mr. Mark Hart. It is a pleasure to welcome everyone to our online lecture. We want to thank to the student lecturer that are willing to make time to join this lecture. We would also like to thank the Dean of Faculty of Mathematics and Natural Science, and will be Comrade maybe, and Mr. Yulia and the team who coordinate this program. We would like also extend a special welcome to our important guest, Mr. Mark, PhD, who will be our speaker today. And in a moment, we'll be presenting an exciting topic about biomarker and exposures of marine ecology. As we know that guest lecture in biology study program is annual activity attended by all biology students in different topics, including in plant biology, animal biology, microbiology, and environmental. Hopefully, this topic will increase our students, as Mrs. Julia said, and increase the knowledge about ecotoxicology, because the toxicology course is not yet in our study program curriculum. We also have hope that this topic will open our minds that there are many things or problem that we should understand and try to solve it, even in small thing. Finally, as mentioned before, that Ms. Vina Rizkawati will moderate today's lecture, and I think this is there will be any question, and in the end of the presentations of Mr. Hartl, at the end of the lecture. Without further ado, I would like to give the time and place to the moderator. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Reni. And all right, maybe now I will let today's moderator to lead this event for Ms. Vina Rizkawati, Master of Science. Time and place and are yours. Thank you.
Okay, thank you, Auriga. Hello and welcome, everyone. Thank you for being with us today. So my name is Vina Rizkawati, and as a part of lecturer in biology program, I will be moderating this session. So for those of you who may not be very familiar with biomarkers and ecotoxicology, today we will learn deeper about that. So you can prepare your note and maybe your pen in case you need to take a note about something during the presentation. So before entering to the main event, I would like to introduce today's presenter. So now Dr. Mark is already here with us, and he will give presentation about biomarkers of exposure in marine ecotoxicology. Dr. Mark, thank you for joining us.
Good morning.
Good morning, Dr. Mark.
In here, good afternoon, Mark.
In here is good afternoon. Okay, so
yes.
Winda, could you please screen share the material for brief introduction for Dr. Mark? Winda, is it possible? Okay, thank you, Winda. Okay, so please give me a few minutes to briefly introduce Dr. Mark to all of the participants. So Dr. Mark is a part of lecturer in the Center for Marine Biodiversity and Biotechnology in Heriot University. And not only that, he is also responsible as director, senior director, and president in some institution. Next, please. And now he is also one of representative in MASTS forum and editorial board member of Fisheries Science Journal. Next, please. Okay, so for educational background, he got his master degree in University of Vienna in 1996. Then he got a doctoral degree in University of Southampton in 2000. And lastly, he got his postgraduate certificate in 2008. Next, please. So in the earlier time of his career, he was a scientist in several institutions. Then before becoming a lecturer in Heriot University, he went to University College Cork as postdoctoral research assistant and temporary lecturer. Next, please. Okay, so his research interests mainly focus on ecophysiology and ecotoxicology of marine organisms, then the effect of anthropogenic activity on community structure, and also he focused his research about pollutants in marine environment. Next, please. And his expertise is mainly about ecology, ecophysiology, and also ecotoxicology issue of marine and estuarine organism. And here are some of the recent projects that he has been working on related to his expertise. Okay, I think that's all for the brief introduction. You can stop sharing, Winda. Okay, without any longer waiting, I think we shall start to the main event, which is the presentation of today's guest lecture. So I will pass this session to Dr. Mark. Okay, so Dr. Mark, time is yours.
Terima kasih.
Sama-sama.
So I'll try and share my screen.
Yes,
can you
please.
hear me okay?
Yes, your voice is clear.
Okay. Let me just, I don't use Zoom very much. I usually use Teams. So I'm just starting the presentation now. And I should be able to share my screen as well. So I think it's this one, yes. So can you see me? Can you see the screen?
Yes, Mark.
Yes, we can see your screen.
Okay, excellent. So in that case, I'll start. So when I first had this discussion with Julia about giving a presentation, we talked about what would be of interest, and she wanted me to talk about my research background and some of the things I've been doing in the field of marine ecotoxicology. What I then decided was that because of Julia's interest in sediments and what we've been speaking about in terms of potential collaboration, I thought I would focus on sediment-associated contaminants. So what I mean by that is, let's see if I can get the next slide. Yeah, so I will talk about biomarkers. I will talk about environmental stress. I will talk about some case studies that will actually be able to exemplify how that works, what research has been done in the past, and also what I would like to do in the future, or what I am doing at the moment. Some of that's been, unfortunately, interrupted by COVID, of course, as was Julia's visit here in Edinburgh 2 years ago. when lockdown happened. So some of the research that I will talk about today is to do with microplastics. Julia and I are also thinking about developing a joint proposal here. I will then talk about PAHs and PCBs, so very common chemicals from industrial processes that are found in sediments. I look at metals in sediments and the potential effect that noise has in interacting with these metals and making them potentially more problematic. And then I'll finish off with two very new projects that we're currently working on, and that is the impact of climate change on biomarker response, and also developing data that can quantify the impact or the potential impact of deep sea mining on the environment of those locations. So that's in a nutshell what I would like to do today. It's going to be a bit of a whistle-stop tour, so I won't be able to go into too much detail about anything, but can you see my cursor, my pointer?
Yes, it's visible.
Yes.
Okay, good. I won't be able to go into too much detail, but you can find papers and relevant details of the data that I'm presenting on my website if you want to get in touch and talk about that at a later date. And I encourage everyone, from the distinguished professors in the audience right down to the students who are interested in discussing this kind of activity. So please don't hesitate to get in touch. Something went beep there. I can't see the chat, so that's nothing for me, I guess. I'm going to, first of all, start off at the beginning, because it's always good to define things, I think. And if we look at toxicology from a classic point of view, there's two things here. There's the hazard assessment. So we're looking to see what the effect is of something, what the potential toxic or pollution-related agent is. And then we want to try and develop a risk assessment so that we can understand the impact that this activity that generates this toxic substance has. And of course, we go through a process of generating a risk assessment. You know that risk is a very simple function in a toxicological point of view. It's exposure time effect. So it's cause and effect, essentially. What the toxicological process tries to do is quantify that effect. And we can do that very nicely in the lab using cell cultures, for instance, that will give us some information about how toxic a substance is. Or we can use animal models. In the aquatic environment, a very popular species, of course, are various forms of zebrafish, but we can use mussels, we can use all sorts of other invertebrates that I'll be showing you today as well. And we can use other more realistic fish models that we find in the environment. And of course, a lot of these classical toxicological studies are performed under very highly controlled conditions. So they have practically no environmental relevance whatsoever. They just tell us how toxic a substance is under the conditions that we're looking at it. That gives us some indication of lethal dose or effect concentrations that we can then turn into policy guidance, and that regulators then can use to make laws, for instance. And you have this similar processes in your country as well. But the issue with this whole thing is that it's very controlled, it's very lab-based, it has no environmental relevance whatsoever. And this is where ecotoxicology comes in, because again, you've got a hazard assessment, you've got a risk assessment, you've got the same causality, risk, a function of exposure times effect. But you have the added complication here of the environment. And as you can see from this little cartoon, the different environmental compartments that we like to categorize to make it easier to model: water, terrestrial, atmosphere, below ground. All these things in reality are not isolated. They don't exist in isolation. They do communicate with each other. And that, of course, adds a major complexity to toxicology, because we have to understand how substances behave, where they move to, do they stay in the water, do they precipitate into the sediment, do they evaporate into the atmosphere, are they transported through the atmosphere, and so on. And all this, of course, adds to that complexity. So ecotoxicology is a lot more complex than traditional toxicology, even though it does draw on a lot of the techniques and a lot of the expertise that we've gained over the years from classic toxicological experiments. Just to show you an example of how complex this might be, hopefully you can see this. This is where I live here. So here in Edinburgh is where I live. And this is the Firth of Forth. So Julie will recognize this maybe. The big Forth bridges are here. And there's a big river that comes down here called the Forth. And it empties into this large bay area. And the catchment area is very large for this river and this basin. And what you see here, these dots, these green dots and blue dots, are wastewater discharge points. Some of them are treated, some of them are untreated. And you can immediately see that this leads to a diffuse contaminant source. So we don't have a point source simply because there is so much going into the river systems, into the Firth of Forth, and that complicates the situation quite considerably. It also leads to complex mixtures of contaminants. So we don't necessarily just, we're not generally not dealing with just one contaminant at a time in isolation like we would in a traditional toxicology test. We're dealing with complex mixtures and how these might interact. And of course, that makes the situation a lot more complex. It also means that we will need to use endpoints of toxicological testing on various levels of biological organization. So we might do something very simple that's very easily done, but not very complex, such as an LD50 test in the lab. And we might go out into the field. We might do some more complex tests along this gradient of complexity here. Of course, with the complexity that increases, the simplicity of these tests and the ease of how we can do that, and the ease how we can do that decreases. So the more complex an experimental design is, the more difficult it is to do, and often also the more costly it is. So in order to get a nice picture of the whole situation, we have to work on all these levels of biological organization, starting from very simple, just fact-finding toxicology tests, right down to some multi-year, maybe multi-annual field studies that try and understand how a certain substance affects a given environment. So that's sort of the context that we're dealing with and that we have to work in. If we look at environmental stressors, there's many of them. Obviously, they can be chemical, it can be noise from shipping or from pile driving. We have a lot of marine renewable energy developments here, wind farms and the like, and all that generates noise during the construction phase at the very least. All sorts of other anthropogenic or human activity. That could be tourism, that could be just, or any other recreational activity, that could be development, anything that you can think of is going to be some form of stress to that environment. And then, of course, more recently, we're getting more and more aware of climate change, how temperature might affect not just the atmosphere, but also the water, and also osmotic pressure or osmotic stress through increased rain, which dilutes the seawater, or increased temperatures leading to increased evaporation and therefore an increase in salinity. And that's, of course, going to have osmotic consequences for the organisms that live in those situations. And because we're dealing with a complex environment, we have to start looking at multiple stressors and how they work together. Are they additive? Do they cancel each other out in an antagonistic way? Are they synergistic? Or they might even be potentiating, which of course means that they are much higher in effect than they would be on their own. So these are all things that we need to look at, and we need to look at this from a multidisciplinary point of view. And that's where the Aquatic Stressors Forum comes in. MASTS, this organization here, Marine Alliance for Science and Technology Scotland, is a umbrella organization in Scotland that contains or includes a lot of universities, a lot of industry, policymakers, government agencies, and Heriot is one of these partner organizations. There's the website here if you want to have a look at it. And within this, this organization, there is a pool of interdisciplinary expertise. And the way it's organized is that there are certain research themes, and within these research themes, we have a series of forums or fora. And one of them that I run is the Aquatic Stressors Forum. You can see it's here under marine stressors. We recently changed the name in order to include freshwater in our activities as well. And this provides a really good basis, as I said, a pool of interdisciplinary expertise, but also resources. So we can go to different partners and use their kit, and they come to us and use our kit. We exchange students, we exchange expertise through other means. We publish together, we put proposals together, and it makes us much more effective than we would be just working in isolation. It also gives us seats on government committees where we can influence government policy by providing data. For instance, the Scottish Government Marine Litter Committee is a committee that develops policy for the Scottish Government to effectively regulate plastics and plastic litter, for instance. And obviously, if you want to influence government policy, you need to provide evidence in the form of data, and that's really all what my kind of research is about, is generating this kind of evidence. So one of the things that I do is monitoring, because it's okay to understand in theory how toxic a substance is, but if it's not actually out in the environment, if there's no risk of or no potential for exposure, then there's no risk. So we do have to monitor. But we don't just monitor for chemicals, We monitor for litter, for instance, the microplastics, as I'll talk about in a minute. But we also monitor for biodiversity and for chemical analysis, so that we know what type of chemicals are out in the environment. When we're monitoring for litter, it's a very time-consuming thing to do. It's very costly, and there's a lot of dependence on. Sorry, I'm going to have to close my window. And there's a lot of dependence on citizen science and the voluntary sector. And there's one organization that you may be aware of, the Marine Conservation Society, that acts not just locally here, but also internationally. And they provide a lot of support for that kind of work. Biodiversity, we need to understand what species should be here, which species shouldn't, which are invasive, for instance. And we can also use species as indicators. We can also use them to transfer them from site to site to look at uptake of contaminants and use them essentially as monitoring tools, a bit like the Muscle Watch program that was developed in the late 70s. Chemical analysis is usually the last resort. The reason for that is that it requires a lot of highly controlled environments to work in, specialized equipment, expertise, and also certification. So it's usually done by very specialized labs and consultancies, often on a commercial basis. And that means it's very expensive. So it's usually the last thing that we do, simply because of that kind of cost. So what we try and do is we try and use more realistic situations where we can actually tell the impact of contaminants on organisms, because chemical analysis alone is not just expensive, it also tends to overestimate toxicity. If you're just looking at the total amount of a substance in sediments, for instance, it overlooks the potential for bioavailability and therefore can overestimate that toxicity. And often the limits of detection are problematic in the environment, because not all contaminants are concentrated in a certain area. They might bioaccumulate, of course, within organisms, but often within the environment, they occur in very, very small concentrations that are often very difficult to actually measure with great confidence. So ecotoxicology, therefore, comes in here. It provides us that hazard assessment, and it provides us with tools and data in particular to actually go about assessing that risk. And one of the tools for this are biomarkers. This is my favorite definition of a biomarker, a biochemical or genetic, cellular, physiological, or behavioral variation in an organism that we can measure. That could be tissue, body fluid samples, or it could even be the whole organism, or even a whole population, if we can measure a change that provides evidence of exposure and or effects, so that's important, of one or more chemical pollutants or radiation in some cases. So that's what we're talking about. We're talking about something that we can reliably measure to try and establish cause and effect. And some of those biomarkers that we use might be physiological. They might be detoxification enzymes such as ZIP1A1, which is a P450 family enzyme that breaks down things like PAHs and from the oil industry, and PCBs from the energy generating industries. And we can use techniques like Western blotting to detect these proteins on a gel from a sample that we take from an organism, for instance. And I'll come to that in a bit more detail in a minute. We can also look at DNA damage. This is something that Julia's been doing in my lab when she came to visit, is using something called the COMET assay, which allows us to detect DNA strand breaks in single cells. So what you see here is fluorescent representation of the DNA of a single cell. And all these bits here are essentially bits of DNA that's broken off and migrated through a gel. And you can measure using this software here the proportion of fluorescence in the so-called tail, the COMET tail, to the head, and that will give you a representation of the level of DNA damage. This, by contrast, would be a perfectly normal cell where there hasn't been very much DNA damage at all. Whereas these two up here, you can see considerably damaged through some kind of agent or stress. There's lots of other biomarkers that we could use. We could use upregulation of certain proteins, heat shock proteins, HSPs, or chaperonins, often called, that are imperative in helping to maintain other protein conformation and the function of those proteins. And they are an indicator of general stress, not just heat, but of general stress. And we can measure those the upregulation of these through various techniques. Or we can look at cellular stress, where we might take an invertebrate cell here. You can see these red stained areas are lysosomes within the cell. We use something called neutral red, this dye here. And in healthy cells, it will initially stay within those lysosomes and only eventually leach out and stain the cytoplasm. But if the cell is stressed, these lysosomal membranes are likely to be damaged, therefore more leaky, and you get a much quicker leakage of the neutral red dye into the cytoplasm. And you can see this in this particular cell here. So these are just some examples of biomarkers that we could use. So I want to spend the rest of the time, if I may, focusing on some of the research I've been doing, using and applying these techniques. And I'll start off with a monitoring case, because I know that Julia is very interested in microplastics. And I'd like to sort of talk a little bit about what we've been doing there. Microplastics, first of all, bits of plastic that are smaller than 5 mils or millimeters. That's the general convention. They could be particulate, like this paint chip. This bar here is a millimeter. So you can see it's just about a millimeter long. But if you look very carefully, you can see bits of blue here on this filter. And they're very, very tiny, down into the micrometer scale, so-called nanoplastics. And that of course makes dealing with these very, very complicated and very, very time-consuming. But they could also be fibrous in nature. There's lots of fibers in the environment coming from various sources. It's no surprise that we're finding a lot of plastic in the environment. If you look at the use of plastic over the last century or so, and we're about here. This is where it's predicted to go. It's just predicted to go up. And the vast majority of that plastic is discarded somehow. Some of it is burnt for incineration, for energy production, for instance. Some of it is still used. Some of it's recycled, but not as much as we would like. The vast majority of it is discarded. And in landfill, for instance, or just gets blown away and ends up in the environment. And this is where citizen science projects come in quite nicely, because you can get people involved to sample sediments or just collect litter from the shore. And you can see here in Edinburgh on a morning, a wet morning, as you can see, just a few volunteers here collected quite a lot of rubbish from the shore, which is quite sad, really. And of course, if you have some storms in the winter, you get even more material that's washed up onto the shore. And what's becoming evident from doing these surveys for many years now is that the vast majority of the items found are some form of plastic. And the Marine Conservation Society has been running these surveys for over 20 years and has a really good data set of marine litter. And you can see that, again, here, just another breakdown of some of their data from 2018. What's really nice about this chart is that you can see that the vast majority are either plastic or polystyrene, but that also a large proportion of the items found are from sanitary waste, so waste that would come through the sewage system. And what's interesting here is that a lot of these materials contain plastic, like wet wipes, for instance, that don't degrade. So they would actually need to be in this section as well. And of course, that would increase the amount of plastic that we're finding. Some extreme cases, of course, of plastic in the environment, floating around. It starts to break up. It doesn't break down in the environment. It breaks up into ever smaller pieces. We refer to these as secondary microplastics. You can see some of these are already in the microplastic range of about 5 millimeters. Some of them are still macroplastic, but they will eventually break up into smaller pieces. And so these are secondary microplastics. And then we have to contrast that with the primary microplastics that we find or used to find in cosmetics. Fortunately, microbeads have been banned in many countries now through a lot of campaigns that were in the press and on television, social media. But they were all, and you'll see that in a minute, these campaigns were possible because of monitoring data that was provided to them. Other sources of plastics through laundry and washing machines, dryers, fibers wash out of clothes. And you can, as I said, you can find these fibers quite regularly in every sample that we take. We find these kind of fibers. Some of them are from clothing, some of them are from ropes. The maritime industry, boats and shipping, more increasingly use synthetic ropes because they're so much stronger than ropes from natural materials. But when they get dry and brittle, they don't snap straight away, but they do shed a lot of these fibers, and we're finding them quite regularly in our samples. Lots of attempts, of course, for laundry devices, filters, and all sorts of catch devices that are designed to retain the fibers from washing machines and from dryers, that they don't actually go out into the environment. To give you an idea of the scale of the monitoring we've been doing, this is Scotland here. So London somewhere down here. This is the Firth of Forth where I am. So this is where we started off sampling around the Firth of Forth. We sampled up in Orkney where we have a campus, and then some of the northern and western islands as well, which are quite isolated. We've done one sampling event on the west coast, Glasgow, a very big city. And I'll just show you a little bit of data coming from the Firth of Forth and from Orkney. And what we do is that we go out at low tide, and you can see here it's still wet, the sediment, and here it's dry. So this is the last tidal line, the high water mark, and that's where we would sample. So we sample some sediment, take it into the lab, extract the plastics, count them, analyze them, and come up with data showing what we found for that particular day at that particular site. And if you want to know more about the processes that are involved there, I'll be happy to talk about those later and send you some papers if you're interested. So if we look at the data from the Firth of Forth, so where I live, we've been doing this for a few years now. This is slightly out of date, but what you can see is that if we separate between particles and fibers, you can see that the trend in fibers is essentially stable. It's not going up or going down in any significant matter. And the same for particles, really. This spike here is a very interesting one. And we think that is to do with an industrial accident that happened earlier that year and is reflected in the plastic particles that we found. So you can see that without this regular sampling, we wouldn't really be able to have a feel for how the amount of pollution is temporarily changing or not. So this is information that we can provide to regulators. If we look up in Orkney, this is interesting here because it's relatively remote. So here, a lot of people live here, big cities, but here very few people live. There is some industry, but generally speaking, it's much more remote and much less going on. And what you can see is different sites around Scapa Flow, this area of the sea here. And you can see that the levels, if you remember, if we go back a slide and look at the scale, we're sort of 0 to 9 microplastic particles or fibers per gram of sediment. If we now go to this slide here, it's the same scale, yeah, per gram of sediment. And we're looking at a very remote or relatively remote location. So what this is telling us is that plastics are everywhere, essentially, even in remote places. And this was quite a stark finding because the press picked up on this. Television came and there was a lot of media hoo-ha. And when things get in the media, the politicians get interested and they start discussing it and start using that data as a case or to make a case for legislation. Now, I'm not claiming responsibility for any of these legislative processes that happened, but what you can see is that a lot of initiatives have been taken by the Scottish government to try and reduce the amount of litter in the environment. So this just shows you a nice way of generating data that actually leads to an impact. I'd like to change a little bit and talk about chemicals in the sediment. PAHs, polycyclic aromatic hydrocarbons, aromatic hydrocarbons, polycyclic aromatic hydrocarbons, sorry, and also polychlorinated biphenyls or PCBs associated with the oil and the energy generating industries. And what we did there was, I used to work in Cork. This is here on the south coast of Ireland. And there's a lot of industry in Cork Harbour. And these two sites here in particular, a big refinery and a big power plant. And we wanted to know how clean the sediment was and how problematic the contaminants that we might find there might be for organisms. And we used turbot as a test species. Turbot is a large flatfish, very nice to eat. It's, as you can see, it's very much associated with the sediment, uses it as a habitat essentially. So it's very intimately connected with the sediment. So what we did is we took sediment from those sites, took them into the lab, and we exposed turbot to these sediments. And we wanted to look specifically for P450 upregulation. P450, an enzyme that sits on membranes, and one of its roles is the essentially phase 1 of detoxification. So it makes things more water soluble in order for them to be further processed by the body. And you can find them very much related with the endoplasmatic reticulum on those membranes, in particular the rough endoplasmatic reticulum with the ribosomes producing that protein. And of course, that protein is available normally as a part of normal metabolic processes, but if there is a case of exposure to a contaminant that can be metabolized by P450s, what you'll get is a significant upregulation of that, that protein. So it will be expressed into greater amounts, and secondly, it will show greater activity. And that's what we're showing here. What you can see is a control. So this is normal metabolic function of the turbot. These are liver samples, by the way. Here, the red is from a control site or a reference site that has no industry. You can see it's relatively over a 3-week period, no real difference in those samples. So the turbot that were exposed to these relatively clean sediment showed no significant change in their metabolic function in that respect. But the 2 sites that were suspected to be contaminated, you can see significant increase in activity of P450 expressed as ERO. And that can be backed up by Western blotting, where we see the 2 sites here, very strong bands of P450 ZIP1A1 with a characteristic size of 50 kilodalton developing in the livers of these species. So we can use this information to go back to regulators and say, look, we think that these sediments are contaminated. In fact, we know for sure they're contaminated. We think most likely this is going to be PAHs and PCBs because of the associated industries. And then chemical analysis can be done much more focused, which saves a lot of money, essentially. So this is a very useful example of how biomarkers can be used in that respect for some organic chemicals. I'd like to talk a little bit about metals and then add noise into the picture to show you that we can't use things in isolation. We need to work on a multi-stressor basis. And what we did here is we exposed mussels to noise, first of all. And what we could see is that some of the stress markers we used were actually affected just by noise, nothing else. In particular, DNA damage, which we think is as a result of increased stress, oxygen consumption, and that leads to more oxidative radicals, which again leads to DNA damage. And we showed that quite nicely in a paper we published a couple of years ago. I think it was 2019. And what's particularly interesting about this work is that if you look at the DNA damage, you can see this is in gills, and this is in blood cells of these mussels. You can see that the controls are very low, as you would expect, but the mussels that were stressed with noise have got considerable DNA damage. We also saw considerable damage in terms of lipid peroxidation. So that means that it's not only the DNA that's being affected here, but also membranes, cell membranes. And that, of course, can have all sorts of consequences with for metabolic processes, maintaining homeostasis at the cellular level. We didn't find too much effect on some of the other oxidative stress markers that we would have expected. What we think here is that the stress levels are not high enough to actually trigger these upregulation in these areas as well. We took that a step further and looked at invertebrates that live in sediments, in this case, a very commercially important species, Nephrops norvegicus, or oh, I forgot what they're called now. There's another word for them in English. And they're very highly prized as food, and therefore are important species in that respect. But they're also very abundant, so they're really easy to collect, easy to grow in the lab, and they're easy to run experiments with. And what we did was we assumed that, well, we know, for instance, that these species live in the vicinity of wind farms. Wind farms are quite noisy places, not just from the production and building, but also from the running, lots of shipping involved with it. And we wanted to look and see how noise and metals together would affect the stress levels. And what we saw was that in particular, the upregulation of metallothionein was much more pronounced in the organisms that are exposed to noise and metals than the ones that were only exposed to either noise or metals. And that's slightly contrast to some of the oxidative stress assays that we used. We couldn't find any major or significant upregulation there, but we're very intrigued about this upregulation of metallothionein. As you probably know, metallothioneins are characteristic for scavenging metals, and this would suggest that the noise and the associated activity is making these metals maybe more bioavailable to the organism, and also adding to that noise stress and the metal stress together gives that that synergistic effect. Very briefly, two of the more recent studies that I've been involved in, we were really interested to see how changing environmental conditions might change the baseline levels of some of the biomarkers that we use. Because if they change anyway because of changing environmental conditions, then we might not be able to, we might not have the resolution in order to be able to detect any significant upregulation or changes based on pollutant exposure. It might be masked by that change in environmental conditions. And of course, in the marine environment, the main issues that we have with climate change are, of course, CO2. So not just temperature that's going up, but CO2 going up, and that leading to localized ocean acidification or a decrease in pH. And of course, that can have all sorts of consequences for not just calcifying organisms, but also for requiring more energy and therefore more stress to maintain homeostasis within cells in order to keep metabolic functions going and working at a normal rate. So what we've been doing is looking again at DNA damage, and again we've been using muscles. And what you can see here on the left are 3 regimes of changing salinity. So one would be just stable, normal salinity, about 32 parts per million. One would be a gradual decrease, and one would be simulation of a rain event, which would of course lead to dilution of the seawater, especially in localized areas where these muscles occur. And now, of course, they are euryhaline. They live in estuarine environments, so they are able to cope with osmotic stress. But then again, the key word here is stress. In order to be able to deal with that, that costs energy, oxygen consumption, leads to oxidative radicals and the like. So we looked at DNA damage here, and what we could see was that if we look at, let me get this right, A is blood, I think. Yeah, so A is are blood samples, and what you can see is here that over a 3-week period, there was very little change in DNA damage. But if we look in gill cells over the same period, we can see that fluctuating salinity does have a considerable impact here on the DNA damage in these organisms. And that suggests that they are simply having to use more energy to maintain homeostasis within those cells. We also looked at different temperatures, not just salinity. And what you can see is here that if we look between summer and autumn, and then up the temperature, we can see that the organisms in either category, so regardless if it's summer or autumn, are experiencing higher levels of DNA damage in their blood cells and in their gill cells, whereas it's not the summer temperatures are much more pronounced in the effect that you're getting, and that might be because the organisms are probably reaching their main or their maximum temperature limit that they can actually deal with without too many problems. These temperatures, of course, are extremes, and they're extremes based on what we can expect in about 20 to 30 years' time if the climate change scenarios keep progressing in the way that they are. So there's a lot more work to do here, and this is just the beginning of this particular project here. And we want to take this further. We want to look at how the baselines of some of the oxidative stress assays that we use, T-bars for lipid oxidation, superoxide dismutase, glutathione, and so on. So big question marks here, and a lot more work to do. And I'd like to finish with the most recent project that I've been involved in, and that is to try and assess the impact of deep sea mining on the marine environment. What you see here is an area of the seafloor out in the Pacific. It's in 4,000 meters depth, and you can see these potato-sized so-called nodules, the so-called manganese nodules. And as the name suggests, they contain a lot of important metals. So not just manganese, but also some metals, rare elements needed for batteries, electronics, and all that kind of stuff. And as these materials become less and less available on land, there is more and more investment and more pressure to mine these marine environments. And we know very little about these environments. And one of the things that we want to do is try and figure out what type of damage these kind of huge collector vessel or collector instruments or tools or devices are going to have on those environments. What we think, obviously, and this is a graphic summary of the project that I'm involved in, is that these devices are going to move along the seafloor and they're going to create a huge plume of sediment. Because there's a lot of metal in these areas, there's a lot of metal in the sediments as well. So we would be introducing metals into the water column. How is that going to affect? So from the sediments, how is that going to affect mesopelagic fish there? So that's essentially what we want to find out. Just to give you an idea where this is Equatorial Pacific, and it's this zone here called the Clarion-Clipperton zone. And all these blotches and colors are licensed areas where people would like to mine. And at the moment, there's a moratorium, but it will go ahead at some stage. So what we are trying to do is to provide evidence for the effect, some quantitative data that can be used to mitigate whatever effects we might find. And one of the things we're doing and trying at the moment is using these baited traps that we put down on the seafloor, and we catch these rat tails, for instance, deep sea fish, and we want to see how their gills are affected by the sediments, for instance, and the metals contained within. So this is a brand new project that has been severely affected by COVID. So all the cruises that we were supposed to do last year and this year have been postponed. So the next cruise that I'm preparing for at the moment is in February, March of next year. And we'll be out in the Clarion-Clipperton zone taking samples and applying these assays. And at the moment, what I'm doing is trying to optimize them for these particular fish species and the sampling conditions, which, as you can see from here, are quite rough because the organisms are either caught or captured within the traps and then have to be brought back to the surface, which essentially kills them. So that in itself is a stressful activity that might mask any effects that we see from the metal contamination. So we have to optimize our tests, our assays, to be able to account for that. And that's what we're doing at the moment. Right, so I'm just going to conclude here, and I hope I've shown you that biomarkers are quite effective in assessing the impact of exposure and measuring it in particular. There's lots of areas that we can develop in, and there's still a huge amount of work to do with lots of questions, and they will vary from site to site, from country to country, location to location, situation to situation. So they're very adaptable and malleable. And one of the issues is, of course, that there's just so much about the natural environment in terms of its complexity that we still struggle to build into our models. And that's where the future research is heading. I mustn't forget to thank my funders. A lot of of this work is funded by external funding agencies, in particular the National Environmental Research Council here in the UK, but also by MASTS, the organization I showed you at the beginning, and of course the European Union as well. And without this kind of thing, we couldn't be doing, or this support, we couldn't be doing this research. And this is something that Julia and I are going to be working on in the next few weeks, is to put together a joint proposal to do some work in this area. And with that, Terima kasih for listening.
Okay, thank you for your presentation, Dr. Mark. And now we are entering to the Q&A session. So if there are participants who want to ask something related to what Dr. Mark have already presented, you can deliver your question in this session. So you can raise your hand and deliver your question directly, or maybe you can do it indirectly by writing it down in the comment section in this Zoom meeting. Okay, you can either use English or Bahasa, and I will help you to translate. Okay, so we have our first question from Mr. Agung. Do you want to deliver directly, Mr. Agung?
I think my question is quite clear, Mark.
Okay, so I will. Winda, could you please screen share
I see. Am I supposed to read the chat? Sorry.
It's okay. We already start the screen sharing, which contains the question, so you can see at your screen. So the first question is from Mr. Agung Sedayu, a lecturer from biology program. The first question is this. So if we want to evaluate the level of toxicity in a certain marine place, for example, in the Bay of Jakarta using biomarker, what do you suggest in the species that we can sample? Do we can use the large pelagic species, or maybe motile planktonic, or maybe the non-motile like seagrass? Okay, that's the question.
Well, as I said, this means that the biomarkers of species we use will depend from place to place. Mackerel, for instance, are interesting, but mackerel, one of the problems with those is that they are migratory. So anything that you might find in the mackerel might not be relevant to that particular location. But what it might show is, for instance, if the mackerel don't come because the situation in the, let's say, in the Bay of Jakarta has become so bad that they stay away. That in itself could be an interesting situation. So mobile species are not so good. And I agree with the second part of your question, that it's usually better to focus on sessile, non-mobile species, because they are more likely to be exposed to these substances chronically, and often they're in very low concentrations. And it's only the chronic exposure that leads to a measurable effect. What was that? Does that help, that answer?
Yes, Mark.
The answer. Okay, so the second question is about the body mass. Is the body mass important when using biomarkers?
Yes, body mass is important. That's why we normalize all our results to body mass. That can either be wet weight or dry weight, and that can be problematic because wet weight is a bit too general. which means that we end up with inaccurate body weights, because many aquatic species are very, very, or have a high water content in the body tissues. If we then dry it, we might destroy the chemical that we're interested in. So we usually get around that by normalizing against protein, for instance, total protein. That's one way of doing that. So yes, body mass is very important.
Okay, Mr. Agung, is that answering your question?
Yes, Mark. Thank you very much. I got a clue. Well, I'm not a toxicologist. I'm a botanist, and I'm stranger in toxicology stuff. So I did a little bit of measuring cadmium and copper inside the mangrove fern, the Acrosticum species, and it turns out that it was quite high in the organs. It was maybe 5 years ago. So if you say the biomarker was very inexpensive compared to chemical what do you call that? Chemical analysis. Yes. It should be a good opportunity for me.
Yes, it can be. Again, it depends on the situation. And one of the issues with biomarkers is that is cause and effect. There are very few where you can be absolutely certain that the cause or the effect has been caused by a certain contaminant. For instance, if you're looking at neurological damage and impairment of acetylcholinesterase, which is an enzyme that hydrolyzes a neurotransmitter, that is specifically inhibited by pesticides, organophosphates, for instance, and some carbamates. So that can be really nicely narrowed down. Most other biomarkers will give you some indication of stress. And the other good example I showed you, I guess, is P450 upregulations. But you wouldn't, I don't think, I'm not a botanist, I'm not sure, but I don't think you would probably have to find some other biomarker within plants to be able to deal with that. Maybe something impairing the photosystem in the chloroplast or something like that.
Yes, thank you, Mark.
Okay, thank you, Mr. Agung. So we are moving to the second question from Mrs. Reni Indrayanti, one of the lecturer in biology program, who is also the coordinator for this program. And she asks about the, so we suppose we already identified that pollutant contaminate the area. What we should do next? Is there any algae, microorganism, or maybe plankton that can be remediate the area that is toxic or contaminated? So it's about the
remediation.
Yeah, bioremediation.
I'm not really an expert in that, but what I do know is, especially here in Scotland, sediments are really only remediated if you really have to do it. And the reason for that is that if you move sediments, you very often, that very often leads to the removal of those, or the release of contaminants into the aquatic environment, very much like I showed with the deep sea mining. So there, as far as I know, there's lots of different remedial strategies like growing seagrass, for instance, or trying to bind the contaminants in plants and other materials. But generally speaking, I'm not too sure from my own personal experience what I would advise you there, because I'm not really a remediation expert.
Okay, is that answering the question?
Okay.
Probably not. Sorry.
Yeah. Okay. Okay. Thank you. That for I have heard that if their sedimentation are contaminated, that we should remove the sedimentation to that area. It is the simple solution for the contaminated area. Is it? True?
Well, in theory, yes. But
okay,
as I say, one of the problems is where I live in the Firth of Forth, there are lots of harbours and ports, and they silt up. And every now and again, that sediment has to be moved and dug up because the ships can't get in. And that is problematic because it releases a lot of contaminants. You get a big sudden pulse of a much higher concentration of substances than you would usually get. So if people don't have to move sediments, they prefer to leave them where they are rather than and try to remediate them in situ somehow. That could be like you suggest with microorganisms or maybe seagrass or algae, or some other way of doing it. Maybe using some novel substance that binds the contaminant much better than the sediment itself does, so that when the sediment is disturbed, is removed, the substance is not released into the water.
Thank you. That's will be interesting to us to make a research about the remediation, maybe in the laboratory, mungkin Bu Yulia and the students. Yes. Okay, thank you.
Yes, that is often the issue. These things work really well in the lab, but the transfer of them into the environment is often very difficult and often financially prohibitive, just too expensive.
Yes. Okay,
so what we have to do really is we would have to put a tax on activities to make it cheaper to remediate than to pollute, basically. Yeah.
Thank you, Bu Vina.
Okay, thank you, Mrs. Reni, for the question and the discussion. Any question maybe from the student? Okay, Auriga, please.
Okay, thank you, Ms. Vina, for the opportunity. So, if I may, actually it's a personal question. So, I kind of read your track record and kind of resume in Google Scholar, sir. And I think I'm pretty sure that it's okay if I say that you are an expert at ecotoxicology. So, my question is, how do you pave your way to be an expert at ecotoxicology? Should I take a master degree or to doctoral degree perhaps, or do you have any tips, sir? Because I'm interested in ecotoxicology, especially in microplastics. I think that's the question. Thank you, sir.
Okay, that's a very good question, and I often get asked that by students applying to do courses here with me. It really depends on what your career aspirations are. If you want to work in industry, for instance, do you want to work in government, a government agency, or do you want to go into research? All three will, at the basis, will have the requirement that you have some kind of, or develop your own expertise in, let's say, ecotoxicology of a certain type of ecotoxicology. That could be through choice of your undergrad program, your biochemistry, your physiology, your chemistry maybe. It could be going on to do a master's and doing projects in that area. And it could also be that you go on to do a PhD to get a higher degree in order to maybe have an academic career where you stay in research and teach. So it really depends on what your career aspirations are, but the basis for all of that and making those decisions is to develop a really good grounding in the knowledge of that field. And it's the same with microplastics as it is with any other contaminant. One of the issues with microplastics is just simply trying to keep up with the amount of literature that comes out. Microplastics is such a big topic at the moment that it's, for one person to just keep on top of the literature is impossible. Daily, lots of papers come out. So one way of getting around that is to join organizations, interest groups, social media groups that deal with these aspects. And you then often get a digest of the information. And go to conferences. I'm going to one next week, for instance. That's one way of doing that. So developing a network is important as well. Going to seminars like this to hear opinions from other people from other places. Yeah, so there's a whole range of things you can do to prepare yourself for a career in this field.
Okay, is that answering the question for Auriga?
Yes, it's pretty answering. Thank you so much, sir.
Thank you for the question. Thank you.
Okay, thank you, Auriga, for the question. So after you finish your undergraduate thesis, maybe you can start preparing your IELTS so you can get the required score for going abroad, for example, and maybe meet Dr. Mark in person. Okay.
Who knows?
Yeah, who knows about that?
Everything's possible.
Yes, true. Okay, from Mrs. Yulia, please. Miss, you haven't unmute your microphone.
Okay. Okay, this is my question about when we read the many articles in this research, we often recognize the term biomarker and bioindicator. Is it the same biomarker and bioindicator, Mark?
No, that's a very good question. A bioindicator usually refers to an organism's presence and absence. So, for instance, the presence of a species that thrives when there's lots of nutrients in the water body might be an indicator of eutrophication. And that might point to things like agricultural runoff and things like that. Whereas a biomarker is something that you can measure within an organism, for instance, the effect on cell membranes or cell metabolism, something like that. So that's a very good question. So those are two very different things, and they're actually often muddled up and mixed up. So thank you for that, Julia.
Okay, this is special one. Special question. I think it is possible if you are as the co-professor for my student.
In principle, everything's possible. It depends on the details, and it depends what the projects are. But I would be very honored to help out as a co-supervisor, if you like.
Yes. Is research shouldn't make the description for the bachelor degree. Yeah. You are co-supervisor and collaborator with biology lecture. It's okay, Mark. Are you available?
I might be. Depends when it is and what the project is. So we can talk about anything and then make a decision based on that. But in principle, yes.
Yes. Okay. Thank you. Thank you very much.
The thesis should be in English, Mrs. Julia.
Yeah.
Okay, thank you, Mrs. Julia, for
the funding format. Okay, thank you very much.
Okay, thank you, Mrs. Julia, for the discussion.
You can take the picture, Auriga, after that.
Okay, so I think there is no other question from the students. So I would also like to give a bit of my question regarding your presentation. So I'm quite new about biomarkers. So I'm sorry if maybe my question is basic. So my first question is about the microplastic. So many people has realized the bad potential of microplastic. Many people more and more aware about microplastic that might be contained in their beauty products, such as toothpaste or any other beauty product. So in your city, Edinburgh, for example, how local people manage to reduce the effect of microplastic, especially in beauty products? Is there any policy regulating the microplastic uses in your local city?
That's a very good question. Thank you for that. Yes, there are. One of the probably the key and most successful campaigns, I guess, was the microbead campaign, and these were these very small pieces of plastic that you have in exfoliating shower gels and creams and things like that. So there was a two-phased process. It started off with a ban of the sale of these. It started off with the ban of the production of these creams with these or products with these particles in Scotland, and then 6 months later or so, they were also banned from sale. So you weren't allowed to import them or sell them. So that's one really good example. And you find it in toothpaste, you find them in, as I said, in gels and the like. So in Scotland at least, and I think in Europe as well, they have banned the use of plastic microbeads in these substances. And that is a direct result of the monitoring and ecotoxicology work that partly myself, but many other colleagues in the UK and Europe and worldwide have been doing.
Okay, so it's quite interesting because in Indonesia itself, we do have policy to ban plastic bag, but for microplastic, we still don't have policy regulating about microplastic usage. So yes, Europe is already a step more advanced than Indonesia. That's really.
Because the second part of the question is what local people can do. I mean, it depends what your circumstances are and what choices you have. If you have the choice, try not to buy something with plastic particles in it. Buy something as nutshells or something, or sand or something like that in it that does the same thing like people used to do. Or when I go to Malaysia, one of the campaigns that is going on there is to try and reduce the use of these single-use packets for food. because, and I assume this is, and I'm pretty sure I remember seeing it a long time ago in Indonesia as well, the use of banana leaves to weave packages for food. So there's a campaign at the moment in Terengganu where I also have collaboration to try and increase the use of these natural products rather than plastic products, because that will help with the amount of litter, and that eventually breaks up into the microplastics that we're concerned about.
Okay, so buying natural products is one of the alternatives that we can do as the local people.
Yes, that would be one way of doing it. Again, it depends on everybody's circumstances and what chances what choices you have. You may not have that opportunities. I don't know.
Okay, thank you very much for the answer, Dr. Mark.
Mm-hmm.
And any question maybe from the students, last chance, before we are ending this main event? Okay, if there are no questions from the participant, I think we are reaching to the end of today's main event. And I would like to say thank you to Dr. Mark for the knowledge he shared today with us. And hopefully it will give our students new insights about the recent problem we face, such as the climate change and also the microplastic that affect organism and our environment. Thank you very much, Dr. Mark.
Well, thank you much for the invitation. So the honor was all mine. Thank you very much.
And as moderator, I would also like to thank all of the participants who have joined today's guest lecture from the beginning until the end of today's event. And also some participants who have given questions and did great discussion during Q&A session. We hope you can learn a lot from today's lecture, and it will encourage a lot of people in here to do more for the environment as a student in biology program. So you can do research further about this kind of tema in your research. Thank you very much. And lastly, I will pass the next session to Auriga, who is the Master of Ceremony of today's event. Please, Auriga.
Okay, thank you, Ms. Vina. All right, so unfortunately, today's event is coming to an end. So before I end this event, I would like to ask everyone to open their camera so we can take the competition for this event. So please, maybe the operator can help us to take the documentation first. Okay. So.
Okay, Auriga, ready?
I will take the documentation for 1, 2, 3. For next slide. 1, 2, 3. Yep, it's done. Thank you.
Okay, thank you for Fabian. So let's close today's event, today's guest lecture by saying Alhamdulillah to show our gratitude to the Almighty God, Allah Subhanahu wa Ta'ala, Alhamdulillah Rabbil Alamin. And I would like to remind all the students from State University of Jakarta, Universitas Negeri Jakarta, to register yourself in the link that has been shared by our committee in the chat section. And lastly, I would like to say thank you for Dr. Hartl as the speaker, Ms. Vina as the moderator, the committees that helped this event to be succeeded. And last but not least, thank you for all the participants that have come to this event. So I'm Aga as today's Master of Ceremony. I want to say thank you for coming, and see you in the next guest lecture. Goodbye and have a nice day. Thank you.
Okay, thank you, Max.
Thank you very much. Thank you to all the
Have a
participants.
nice day, Mark.
Okay, I would like to leave in the Zoom. Thank you very much for your all people to attend in the guest lecture. Bye. Thank you.
Goodbye. Goodbye.
Goodbye.
Very much.